So in this section, we are going to talk about the process of absorption. Now, what is absorption? What do we say about the word absorption when it comes to digested products? Now, absorption, it basically refers to the movement of small molecules. Digestion has produced small molecules. Molecules of what? Complex biomolecules. Huge, big, complex biomolecules like proteins, carbohydrates, they are all converted into their simpler forms like amino acids. They are converted into their simpler forms like monoglycerides, fatty acids, like that, right? So these small biomolecules, they have to be go through the absorptive epithelial cells of the intestine. Now, if you see, this is our intestinal lumen. This space here, we will call it as the lumen of intestine. Right, so this is the space of the lumen. Means if, let us say, I'll use the same color to draw it. So this is the intestine, suppose, because intestine is a tubular structure. This is the intestine. Now these areas which I'm highlighting with the pink, they are the wall areas. They are the wall areas, the wall of the intestine, which contains these similar compositions, the serosa, the musculosa, and the submucosa and the mucosa layers, just like in the what we have seen. This space here, this is called as the lumen of intestine. So all the digestion, all the preliminary digestion that happened, it happened in the lumen of the intestine. All the breakdown of proteins, carbohydrates, everything that we have seen in the previous videos, all of that is happening here in this space, in this lumen of intestine. Okay, now what happened? Whatever small molecules that are formed in this lumen of intestine, they have to enter into the walls. Okay, so you have the small molecules like this. Let us say these stars are those small molecules, small biomolecules. We'll use another color also. So we are using some green molecules here. Okay, so these are all the small molecules which are formed as a product of the digestion. Now these molecules, they have to enter into these epithelial surfaces. You can see these epithelial surfaces, right? So they have the microvilli. That's why this intestine is called, these are the villi, right? These are the villi. Remember, and each villi has got the microvilli made epithelial cells, which are called as the brush border cells. Border cells, right? And you also have the intestinal crypts, which also produce some glands, and all the digestive juices, intestinal digestive enzymes, whatever are there, they are coming from these cells also. So these small molecules, what they have to do is they have to first pass through the epithelial cells. Now what is this layer? This layer is called as the mucosa. Sorry, now this layer is said to be the mucosa layer. Right, now this here is the submucosa. This layer is here the submucosa. Right, let me just take the black color and highlight it a little more easier. Submucosa. Okay, and this here is the muscularis layer. Muscularis, remember the wall layers, and this is the serosa layer. Serosa, I'm writing it here even, muscularis. So these are all the submucosal layer. Submucosa is highly vascularized, right? And this is the mucosal layer, right? So these substances, they have to enter into the epithelium cells. They have to enter into these epithelium cells via the villi, and from there they have to enter into these blood vessels and lymphatic vessels. Can you see here this red one? This red one is the arteriole. Right, the green one is the venule, and this the blue one is the venule, and this green color one, which is called as lacteal, is nothing but lymph vessel. It is nothing but the lymph vessel, which is called as lacteal. So the substances have to enter through the epithelium into the blood capillaries or into the lymphatic vessels. Either they will enter into the blood capillaries or they have to enter into the lymphatic vessels. And through the lymphatic vessels and the blood capillaries, they are sent to various locations. So this process is called as absorption. Okay, so there are three ways by which this absorption can happen in the small intestine. The first method is called simple diffusion. Simple diffusion. Right. The second method is called facilitated transport. Facilitated transport. Right. And the third method is active transport. So these are the three ways by which your absorption process occurs in the small intestine. Now simple diffusion is completely based on concentration gradient. So substances move from their region of high concentration to the region of low concentration. So region of high concentration to low concentration. So it's just that downflow movement. Facilitated diffusion, you need the help of transport proteins. You need the help of transport proteins which help movement of substances across the membrane, right? And you will also need the help of some other ions. Active transport requires the movement of substances mainly using energy. Because active transport is against the concentration difference. It is not like simple diffusion where you are moving from high concentration to low concentration. In fact, you are moving into the opposite side. You are moving from the low concentration region to high concentration region. So you will need active transport. We have discussed all of this in the chapter transport in plants even more detailedly. If you just refer to them, you will get a more clearer idea. So in this diagram, you can clearly see the various transport processes, absorption and movement of various substances throughout in the lumen and then followed by the epithelium. So this is the lumen of the intestine. This is the epithelial cell of the intestine. And here will be the blood capillaries. And here somewhere you will have the lymph or the lacteal. Okay, so let us see the movement of substances. First, we are going to talk about the simple sugars. How they are going to move. So, absorption of monosaccharides. So monosaccharides, they are always present in the form, if you remember and go back, after digestion you have glucoses, galactoses and fructoses, right? So glucose and galactose, they move with secondary transport with Na, means they are going to use the co-transport method, means they will move along with the sodium ion. Along with the sodium ion, the glucose and galactose will come inside, and from there they will further move into the blood capillaries. Okay, fructose on the other hand comes in with the help of transporter proteins. There will be a group of transporter proteins will help in the movement of fructose, right? So sometimes small amounts of glucose are absorbed by simple diffusion, but majoritively fructose, which is in fact a monosaccharide found in our fruits, they are transported via our facilitated diffusion. So this is with respect to the glucose, galactose and fructose, and finally from there they will enter the blood capillaries. So they will enter into the blood capillaries. With respect to amino acids, they either move in the active transport way or they move in the secondary transport way. So what is this? Normally, if you have small amounts of amino acids, if you have a very small amount of amino acid, they can enter into the epithelial cell via simple diffusion. That is the concentration difference, right? But if the amino acids if their number is more, either they will take the help of the co-transport with sodium, just like what glucose is taking. So these transporters, they are called as glucose transporters. Glucose transporter, which has a symbiotic movement with sodium, right? So the same glucose transporter is used to transport amino acids also into the cell. Otherwise, they can also use the movement of energy, that is, they'll take the active transport. If they are in the form of dipeptides or tripeptides, they'll use ATP and they will move from there into the epithelial cells. From there, they also move into the blood capillaries. But when it comes to fats or fatty acids, the things will change. Absorption of fatty acids is a little different. Okay, now if the fatty acids are short chain fatty acids means what? They have less than 12 carbon atoms. They have less than 10 to 12 carbon atoms. They are called short chain fatty acids. So they directly enter into the epithelial cell via simple diffusion, and from the simple diffusion they also enter into the blood capillaries. But if it's a long chain fatty acid means what? More than 10 to 12 carbons are there. It's a long chain fatty acid. Or if it's a glycerol, that means they have come from that micelles. Remember, triglycerides undergo emulsification to form a micelle, and that micelle is acted upon by the pancreatic lipase to make them into fatty acids and glycerides. So this is coming, you can say this literally, this structure is coming from emulsification. Okay, so when this kind of thing happens, whatever long chain fatty acids and monoglycerides are there, they will first enter by simple diffusion into the cell, and in this cell they revert back to their triglycerides. They form a structure called as chylomicron, and this chylomicron, instead of going into the blood capillaries, they are going into the lacteal cells or the lymphatic vessels. So how that is going, we are going to see. So in this diagram, we can clearly see that the fatty acids which are come from and monoglycerides, they are all entering into, after emulsification, they formed into the micelles which got digested, and they are entering into the intestinal cells of the mucosa intestinum. So this is the intestinal epithelial cells. Intestinal epithelial cells. Okay, now once inside, what happens is these fatty acids, they enter and they link, they join together to form the triglycerides. Then these triglycerides, as you can see, they combine with they enter into the Golgi complex. In the Golgi complex, there will be a lot of proteins, right? So these Golgi proteins and triglycerides, they form aggregates, and these aggregates are called chylomicrons. I am writing the spelling here. They are called as chylomicrons. What are chylomicrons? They are nothing but aggregates of triglycerides plus proteins. Right, so these are called as the chylomicrons, and these chylomicrons now leave the cell, and they are leaving here. Can you see? They are leaving by a process, and this process is called exocytosis. They move out by a process called exocytosis, and what they do, they, instead of entering into the blood capillary, they are entering into the lacteal. So in this way, they have entered into the lacteal. Okay, so they leave the absorptive cell via exocytosis, and then they enter the blood capillary. Instead of blood capillaries, they enter into the lacteal. Because why are they entering into the lacteals? Please remember, chylomicrons, they have comparatively, they are larger in size. They are larger in size with respect to other amino acids and carbohydrates, right? So the lacteals or the lymph, they have bigger pore. They have the bigger pore size. The pore size for the capillaries are more in the lacteals, and that's why they enter in the lacteal. And from the lacteals, they are transported via the lymphatic vessels into the thoracic duct, and then they enter into the blood capillaries. So this is the this is the absorption of fats. This is specifically the absorption of fat in the small intestines via using the lacteals. So as we have discussed, in the villi, you are having the blood capillary, which is the arteriole, you are having the venule, and you are having the lacteal. And we have seen that short chain fatty acids, amino acids, monosaccharides, they enter into the blood capillaries. And from there they will enter into the venule. Through the venule, they will get circulated like this. They will be circulated in the venules. They enter into the veins, and they finally join the hepatic portal vein. These are the part of the portal system, right? So they enter into the hepatic portal vein, and these enter into the liver. In the liver, these short chain fatty acids, amino acids, monosaccharides, they are first selected, or they first are screened through by the hepatocytes. Hepatocytes are going to see if those which are there, they are useful, harmful, etc. And if the hepatocytes or the liver cells does not remove it, if it does not throw them away saying they are not good or they are not needed, then these structures, the short chain fatty acids, the amino acids, the monosaccharides, from the liver, they enter into the general circulation of the body for the further absorption. But if you see these are the chylomicrons, right? These are the chylomicrons, the long chain fatty acids, right? So these chylomicrons, what they have done, they have entered into the lacteals. They have entered into the lacteal. So from the lacteal, what they are doing, they are entering into the lymphatic vessel. From the lymphatic vessel, they will enter into an area called as thoracic duct. Through this thoracic duct, they will enter into blood and blood tissues, peripheral tissues for circulation. So through the thoracic duct, you can see where it is going. The thoracic duct is directly going and joining into the left clavien vein. So when it is moving like this, the fatty acids, they will enter into the peripheral tissues, and the other blood capillaries. So this is the difference of movements of sugar, amino acids, short chain fatty acids, and that of the long chain fatty acids. So this completes the process of absorption of digested food material. 90% of the digestion happens in the small intestine along with the 90% of the absorption. Only 10% of the absorption occurs in the stomach and in the large intestine. In the stomach, mostly few vitamins and vitamin B12 is absorbed. In the large intestine, whatever digestion, bacterial digestion that is left over, whatever drugs that are left over, they are going to get absorbed in the large intestine. All right. So this is the process of absorption.
For more content, like, share and subscribe to www.i-win.in.